Assalamualaikum and hello to everyone who is watching. Our group will be presenting on understanding the molecular regulation of Hyphae growth in Candida albicans. Before I start, my name is Nurul Ashikin and on my part, I will present on Candida albicans and the difference between each forms. Candida albicans is a benign member of microbiota, normally a harmless fungus that live on our skin and mucosal surfaces such as mouth and vagina. However, there is also an opportunistic fungal pathogen which can cause mucosal infection when our immune system is disrupted, such as oral thrush and vaginal candidiasis, but also can cause a life-threatening infection to susceptible patients. One important characteristic of Candida albicans is its ability to switch between three growth forms: yeast, pseudohyphae, and true hyphae. The switching is triggered by environmental conditions and play an important role in its ability to cause disease. Now, let's look at the differences between the growth forms of Candida albicans. First is the yeast form, a round or oval, a unicellular organisms that reproduce by budding. This form is non-invasive in the human body. The yeast form is found when the immune system is healthy. In lab studies, the yeast form is typically induced under acidic pH and at a lower temperature of around 30 degrees that favor budding growth. Therefore, its main role is to live harmlessly on the skin and mucosal surfaces. The pseudohyphae form is a long-elliptic and multicellular organism, consists of elongated cells that remain attached after budding with constriction between adjacent cells because the daughter bud remains attached to the mother cells after budding and forming a septa with a minimum of 2.8 micrometer in size. In lab conditions, pseudohyphae are induced at around pH 6 and under nitrogen-limited conditions. In the human body, nitrogen is mainly obtained from amino acids. When available nitrogens become limited, Candida albicans sense this nutrient stress and switch to the pseudohyphae form. Adaptation helps the fungus search for nutrient and initiate tissue invasion. Finally, the hyphae form is a tubular and multicellular organism, consists of long tube-like filaments with parallel sides and no constriction between cells. Unlike pseudohyphae, the septa in true hyphae do not produce the pinched appearance, giving the filaments a continuous tube-like structure about 2 micrometer in size. In lab condition, Hyphae are induced at neutral to alkaline pH at temperature of 37 degrees in the presence of serum and acetylglucosamine or embedding matrix. In the human body, similar conditions occur during infection. These host signals simulate the formation of true hyphae. The hyphae form is the most invasive because it enables Candida albicans to penetrate host tissue, causing severe invasive infection.
Today, I will explain how environmental signal trigger hyphae growth in Candida albicans. There are 6 major environmental signals involved. The first signal is temperature. When Candida enter the human body with a temperature 37 degrees Celsius, is it detected by HSP90 or heat shock protein 90, together with responsive signaling pathway. As a result, the CMP-PKA signaling pathway is activated and promote the genes that are responsible for hyphae development, allowing the Candida to switch from the yeast into the hyphae. Number 2, we have pH changes. The Candida can detect pH change by the RIM101 pH sensing pathway. In an acidic environment, the Candida remain in the yeast form. However, when the environment become natural or slightly alkaline, the RIM101 become activated. RIM101 then induce the specific gene that promote hyphae formation, helping Candida invade the tissue more effectively. Number 3, we have serum and nutrient availability. These nutrients are detected by nutrient receptor, an enzyme called adenylyl cyclase, known as CYR1. The activation of CYR1 will increase the level of cAMP inside the cell. High cAMP level will activate protein kinase A or PKA and stimulate hyphae growth by activating EFG1. The fourth environmental signal is elevated carbon dioxide concentration. Human tissue contain approximately 5% carbon dioxide. This increased carbon dioxide concentration is detected directly by CYR1. The activation of CYR1 again increase cAMP production, which activate the cAMP-PKA signaling pathway. As a result, hyphae growth is promoted. The fifth signal is hypoxia or low oxygen level. Certain part of the body, such as a deep tissue, contain low oxygen concentration. The Candida detect hypoxia through oxygen responsive regulatory protein, low oxygen alter fungal metabolism, and activate transcription factors such as EFG1 and UME6. Both of these transcription factors involved in hyphae elongation and maintenance. And the last one, the final signal comes from direct contact with host cells. When Candida attached to epithelial or extracellular matrix, it detects this interaction using cell surface receptor. This activates both the MAPK signaling pathway and the CMP-PKA pathway. This pathway then activate transcription factors such as EFG1, CPH1, and UME6 and stimulate hyphae formation.
Let's move on to the transcription factors regulating hyphae. So, a transcription factor is a specific type of regulatory protein that binds directly to the specific DNA, either to turn the genes on or off. As for hyphae regulation, it is divided into two, which are positive regulator and negative regulator. And each positive and negative regulator are also divided into master and supporting. We look first for master positive regulator. We have EFG1, which is a central transcription factor promoting hyphal growth through the CAMP-PKA pathway. CPH1, a major activator in the MAP kinase pathway for filamentation. UME6, a key regulator that maintains and extends hyphal growth, and it also helps to maintain the expression of hypha-specific genes. As for BRG1, the last one, it is a master transcriptional regulator that are required for biofilm formation of Candida. As for master negative regulator, we have TUP1 and NRG1. TUP1 is a major global repressor that blocks hyphal-specific genes and it prevents the hyphae from forming. NRG1 works together with TUP1 to keep the hyphae genes turned off when conditions are not suitable for hyphal growth. So, both TUP1 and NRG1 must work together as a TUP-NRG1 complex to complete their works. These are the list of supporting and negative regulators. Generally, these supporting regulators work together with master regulators, either to activate or inactivate the hypha-specific genes and maintain the filamentation and also fine-tune hyphal growth in response to different environmental conditions. If you are wondering what is the differences between master and supporting regulator is, one of the differences is that master regulator is like the central of all regulator, while for supporting regulator, they need help from the master regulator to be activated and do their works.
Here are the 4 pathways that involve in Candida growth. Fungus uses this pathway to react, switch form, and to survive. On this slide, we can break this down into 4 main pathways. First, let's have a look at the top red box, the pH response pathway. This pathway is activated by 2 specific conditions, such as alkaline and neutral environments. When surrounding pH shifts away from acidic condition, this pathway helps the fungus to change its cellular functions to match with a new environment. Next, moving to the purple box on the left, we have this CAMP-PKA pathway that stands for cyclic adenosine monophosphate protein kinase A. This pathway is a major hub for sensing nutrients and host signals. It triggered by 4 things: amino acid, carbon dioxide, GLC and AC, or stands for N-acetyl glucosamide, a sugar found in cell wall, and last but not least, serum. When this pathway is turned on, the fungus become more aggressive to grow. Now, look at the green box on the right, the CEK-MAPK pathway, also known as mitogen activated protein kinase pathway. This pathway is highly sensitive to physical stress and structural changes. It activated by embedded matrix, cell wall damage, low nitrogen, host body temperature at 37 degrees Celsius, and again GLC and AC sugar. When fungus feels restricted or damaged, this pathway helps in adapt, change shape, and survive. Finally, at the bottom in the blue box, there is HOG-MAPK pathway. This pathway is dealing with environmental stress triggered by osmotic and oxidative stress, like when the fungus under attack by host immune system or facing extreme pressure changes. HOG-MAPK pathway acts as a survival shield to protect the cell. Moving to the signaling pathway products, there are 2 types of pathways. The first one, major pathways, and the second one, supporter pathways. Let's have a look at the major pathways first. In major pathways, there are CAMP-PKA pathways and also CEK-MAPK pathways that involve. Let's have a look at the CAMP-PKA pathways first. The end product of this pathway is UME6 that functioning in hyphae growth and elongation, specifically for morphogenesis, glycogen synthesis, mitochondrial activity, and also energy metabolism. Next, in the green box, there is CEK MAPK pathways. The end product of this pathway are CPH1 and ACE2 enzyme. These two enzymes are functions to regulate hyphae transcription in cell division, morphogenesis, and biofilm formation. For supporter pathways, there are two pathways that also involve. The first one in the red box, pH response. For end product of this pathway is RIM101 for regulates pH control, morphologic transition, and virulence. Also UME6 for hyphae growth and elongation. And lastly, in the blue box, HOG MAPK pathway that activate HOG1 for biogenesis that controlling shape-shifting of growth.
Okay, next, let's move on to the mechanism of hyphae growth. Our journey begins when the Candida albicans sense an environmental signal. So, inside the host, it feels the warmth of 37 degrees sensor serum and detect the change in pH, also CO2. So, these signals are interpreted by the CYR1 to activate the CMP, PKA, CEK, MAPK pathway. Simultaneously, inoculation releases cells from the carcinosol, a quorum sensing molecule that normally inhibits hyperinitiation. So, by this signal trigger the activation of the hyperspecific genes, it clear out the master repressor NRG1 through the transcriptional downregulation and protein degradations. So this often an opportunity for positive regulatory like UME6, HGC1, and BRG1 to activate the hyper transcriptional program. The cell immediately shifts toward the cell polarization, where the cell stops flooding and begins polarized growth with the help of the HGC1 CDC28 complex, directing growth to the hyper tip rather than at the entire cell surface. So instead of growing all of like a balloon, it focuses all its energy on one tiny spot. To support this, the cell set up the actin organizations, long cables that act like a train tracks. So imagine motor proteins like MYO2 carrying a cargo, like basically a secretory vesicle filled with the cell wall budding enzymes, along the track to supply the center at the tip that call speeds and copper. So this focus delivery leads to a tip elongations where these vesicles are then polarized and fused with the plasma membrane. So it goes into a long straight tube with a parallel side. As the tube stretches out, the cell needs to manage its internal brain. It begins DNA replication to prepare for the division of the genetic material in the new hyphal compartments. The nucleus undergoes mitosis between the growing filaments, so this process is highly regulated to ensure that as the hyphae become multinuclear, each compartment receives a nucleus. Crucially, the cell performs a nuclear migration. Unlike yeast, where the nucleus stands near the neck, hypernuclei must migrate into a growing tube. If the signal for this migration are broken, the cell become confused, leading to a multinucleated abdomen large cell. Next, the cell marks its division point. In true hyphae, septing ring formation is unique because it happens inside the new tube rather than at the junction with the mother cell. This ring serves as the scaffold for the division machinery. Following mitosis, an ectomizing ring assembles at the site of the septum ring. This ring will eventually contract to facilitate the physical division of the cytoplasms. The cell then begins a chitin deposition at the septum ring site to form the primary septums. Proper chitin deposition is essential for the structural integrity of the hyphal chain. This will lead to the septum formations, where cell separation is actively inhibited in Hyphae. The HEC1-CDC28 complex phosphorylates EFT1, which represses cell separation genes that would normally allow these two cells to detach. Even though a divider is built, the cell remains as a connected hyphal compartment with tiny pores between them. Unlike yeast, hyphae do not fully separate because a protein called HGC1-CDC28 turns off the cutting genes, where the SAP7 help stabilize this ring and maintain the connection between the compartments. Finally, through continuous hyperelongation, the fungus maintains this long multicellular chain by remodeling its DNA to stay locked in this invasive stage. Achieved through the chromatin remodeling, BRG1 recruits HDA1 to reposition nucleosomes at the promoter. In host conditions, hypoxia or CO2, UME6 is stabilized, creating a feedback loop for the indefinite growth.
Moving on to the core process of our topic, which is hyphal elongation. Before any physical elongation or vesicle transport take place, the fungus must find response to its environment. External cues like changes in temperature, nutrients, or contact with the host surface will trigger internal signal pathway. This activates master molecular switches, specifically CDC42 and RAS1. These switches cluster at the tip will establish cell priority, effectively drawing the blueprint for where the cell wall need to grow. This is what recruits the actin cables and activate the secretory machinery as we see in the next step. To understand how the fungal mycelium is achieve its directional growth, the processes begin at the step 1 inside the cell, at the Golgi apparatus, where the apparatus will package up wall-building enzyme and membrane materials into a tiny transport bubbles called vesicles. Then moving on to the step 2, which are SEC4 activation. On the surfaces of this vesicle, a molecular chain reaction will occur. YP331 will activate SEC2, which in turn activates SEC4 to form a complex. This regulatory complex is recognized and bound to the myosin motor protein, which then hinge the vesicle onto the actin filament to deliver the vesicle to the tip. Once the vesicle tagged with the activated SEC4, we move to the step 3, which are actin and myosin motor protein. Fungal cells cannot rely on a simple division, so they will use a dedicated tract called actin filament that are stretched straight to the hyphal tip. And myosin motor proteins, specifically MYO2 and MYC1, will drag the SEC4 tag vesicle to move forward. They will act as a transporter to move the vesicle to the hyphal tip. Before reaching the hyphal tip, the vesicle will reach a region called sphincter copper, located just beneath the apical membrane. The sphincter copper is a dense, highly dynamic cluster of incoming and outgoing vesicle. Its main function is to act as a vesicle supply center. It gathers thousands of these transport vesicles, keep them organized, and play a dual role in managing both exocytosis and endocytosis. Exocytosis is a process of the outward secretion of enzyme and membrane materials, and endocytosis is a process of recycle excess membrane backward to keep the growth zone focuses. Finally, the last step, which we reach the exocyst in crescent. Once the vesicle exits the sphincter copper, they need to fuse with the outer plasma membrane. So, the exocyst is an egg protein complex will catch the vesicle coming out the sphincter copper and help them fuse with the plasma membrane in a crescent shape. And lastly, the vesicle will release all the enzyme, which then will build a new elongated cell wall.
I will explain about role of hyphae in pathogenesis and immune evasion. There are 5 main roles of hyphae. The first role is tissue invasion. After fungi enter body, they grow into hyphae. So hyphae invade tissue in 2 ways. Firstly, in mechanical penetration. Hyphae grow continuously from their tips, pushing between epithelial cells and penetrate deep into tissue. Secondly, by releasing hydrolytic enzymes such as protease, lipase, and phospholipase. So this enzyme break down protein, fat, and connective tissue, making it easier for fungus to spread. As tissue damage occurs, body will release inflammatory cytokine which cause inflammation to occur. The second role is biofilm formation. Biofilm formation begins when fungal cells attach to surface using adhesion protein. Then, they grow into hyphae and produce a protective matrix made of beta-glucan, protein, lipid, mannans, and extracellular DNA. This matrix acts like a shield. It blocks antifungal drug and prevents immune cells from reaching the fungi. Because of this, fungal infection become difficult to treat and become chronic. The third role is escaping macrophage. So macrophage usually kill fungi by engulf them through process called phagocytosis. However, some fungi can survive after engulf. So how will they survive? They had a few tactics, but I will simplify it. So firstly, when macrophage engulf these fungi, they continue growing into hyphae until macrophage itself rupture and burst. Fungi also can produce ammonia, which reduces acidity inside phagosome and making digestive enzyme less effective. Other than that, fungi produce antioxidant enzymes such as catalase, and superoxidase dismutase to neutralize reactive oxygen produced by macrophage. This tactic allows them to survive and continue spreading. The next one is immune evasion. It is related to three of the rules I mentioned before because hyphae evade immune system by avoiding recognition, surviving inside immune cell, and forming protective biofilm. So this allows the infection to spread deeper into tissue. For example, during infection, when yeast change into hyphae form, enabling it to invade vaginal lining and evade immune system. This immune evasion makes it difficult for body to completely clear the infection. Finally, after all these rules, mechanism combined, it will increase the pathogenicity of fungi. Means that the ability of microorganism to cause disease increase. So why it can cause disease? So look at its role. Tissue invasion damage tissue organ. Biofilm formation make it resistant to treatment. And escaping macrophage allow fungi to spread. Immune evasion help fungi avoid this destruction. Together, this mechanism cause infection to become more severe and difficult to treat, especially in immunocompromised patients. That's all from us. Thank you.